How are you? I want us to begin on our discussion. Introduction to Food Microbiology. So, Introduction to Food Microbiology, our introductory lecture will entail um my class, of course, is hostel catering and nutrition class, and this is basically what you're going to learn today. So, when you're talking about food microbiology, basically you're talking about microbial organisms that may destroy food, may cause food poisoning, and may cause food infection. So, and then I will ask you, when good goes spoiled, what will you do? Of course, you can think about that. What you will do once you realize that the food has gotten spoiled. So there are so many things that comes into being when you're talking about food microbiology. And of course, food microbiology is the study of microorganisms that inhabit, create or contaminate food, and the study of microorganisms causing food spoilage. So we can't just talk about those that just lives into the food, but the impact that it may cause the human being once that food that has been inhabited by the microorganism may be able to cause the outcome that may come with it. So, what is the importance of food microbiology? There are so many importance that you might realize of you having to study this course, but one of the importance is that the study of microorganisms causing food spoilage, fermentation, and yeast production is essential in food production. So basically, what this is saying, it says that uh, you when you study food microbiology, you'll be able to know that they cause food spoilage. These mechanisms cause food spoilage. They also cause they may enhance and help you to help in fermentation. And also, of course, you realize that yeast production, yeast is also a very important microorganisms in food production. For example, used seed yeast in raising or raising bread. For example, bread, when you want bread to have some air in it so that it can be baked nicely, that is what is used. And I think that is a fact that all of us know that yeast is commonly used in baking, and yeast is also used in fermentation, and yeast is also used in other ways of food production. So, role of microorganism found in food, spoilage, of course, one of those role is spoilage. It plays a very big role in spoilage of food. That is one. And the second one is fermentation. Fermentation is another role played by the microorganism, because most of the fermentation and all the fermentation only happens in the presence of microorganism and specific microorganism. Not all microorganism can cause fermentation. For example, I'll give you time to think which type of food requires fermentation. Maybe you can say that. I'm giving you 1 minute to think about the food that requires fermentation. Then we can talk about food production. And of course, food production here, you can talk about baking. You can also go back to fermentation as well, because that is how some of the food are produced. And when you're talking about baking, there are so many types of food that can be baked. And when you're talking about fermentation, of course, wine. I don't know whether wine is food or not, but it's taken into the mouth and it doesn't cause so many big problems if taken in a considerable amount. But think about that as a way of producing food. Sausages and so many other food that are fermented. And and then microorganisms are used in brewing. Of course, brewing is alcohol, and maybe it can return us to back to fermentation, but brewing as a way of making that drink. When making, I think I might be repeating myself. Baking, pickling. I'd be very much interested for you to go and read around pickling as a method of food production and other food making processes. They also used to control the fermentation process in the production of cultured dairy products such as yogurt, cheese, and the cultures also provide flavor and aroma and inhibit undesirable organism. So, do you think bacteria can be consumed as or for prophylactic measures? Do you think there's a way you can consume the bacteria in a way that it may help you in one way or the other? I want you to think about certain food that can be taken with such bacteria, and why do you need to consume such bacteria? So another task I'll be able to give you is to be able to get some notes, make notes from this presentation, this slide alone, on the role of microorganism in food. And you give me around 10 based on this slide alone, so that you should also think outside the box. For example, I'll give you one hint. Wine making as a way of making food, which microorganisms is used and essentially mostly common used in wine making? Let me give you another example. Baking. Which type of organism is normally used in baking? And the last one is pickling. And I want you also to think about other ways on which the microorganisms play a very big role in food. And remember, the role can be positive and can be negative. So, primary sources of microorganisms, we have soil and water, plant and plant products, food utensils, GIT of animals and humans, food handlers, animal hides, air and dust, and animal feeds. So these basically are primary sources of microorganisms for you to be able to understand for the day. Of course, I'll give you a task. I'll want you to look at the soil and water. How is it a primary source of microorganisms in food? And then plant and plant products. How are plant and plant products becoming primary sources of microorganisms? When you look at food utensils play a very big role as far as microbial contamination of food is concerned. So I'll give you, when you're looking at GIT of animals and humans, these are very important primary sources of microorganisms in food. And it will be very important that you understand how the GIT of animal and human being can be a source of microorganisms in food, looking at them as being source of course, GIT of animals. Of course, some of us consume the GIT of the the other part of the animals that might be contaminated. When you look at the GIT of humans, of course, here you're talking about fecal-oral transmission, and this is very important, especially when you're talking about food transmission. Food handlers, you can ask who is handling food. How hygienic is that person who is handling that food? And of course, one of the ways that we always emphasize is that food handlers can be actually a very big problem, because if you're handling food and you have not washed your hand nicely, you have a running nose, you are diarrheaing, then you might transfer some of the microorganisms into the food. When you're looking at animal hides, for example, let's look at a way you are milking your cow. Then when you're milking the cow, then what do the cow choose where they sleep? We say GIT of the animals can be a primary source of microorganism. So those animals defecate everywhere. So if you touch those animals as you milk them, then you might contaminate the milk. I want you to think about some of the microorganisms that can come up from animals, and that is zoonotic infection. And you can write them down. Some of the common microorganisms that can be transmitted from animal to human being. And when you're talking about air and dust, basically you're talking about leaving your utensils open, you're leaving your food uncovered, and those dust contain some of the microorganisms. And air also is having a lot of microorganisms. For example, if you leave a piece of bread on the table tomorrow and the following day, you'll find that it has been spoiled. So that means that in the air, we have a lot of spores for the fungi that are able to be deposited in the bread, and then the following day you'll find that the fungi has grown. So animal feeds also, when we're talking about animal feeds, we talk about basically food that has been kept for long. Maybe it has been contaminated with insect, and probably it might contain some of the microorganisms that are very important as far as foodborne illness is concerned. So the causes of food spoilage, we have intrinsic parameters and extrinsic parameters. Intrinsic parameters, these are parameters of plant and animal tissues that are an inherent part of the tissues. And these parameters are as follows: the pH, moisture content, oxidation reduction potential, nutrient content, antimicrobial constraint, and biological structures. So the biological structures will basically looking at how is the food initially when harvested. For example, maize. Maize, before you harvest the maize, it is covered. And then when you're talking about it being threshed, then you have the seeds, the maize seeds, which can be crushed to make ugali and porridge. But you also have motherkoi. So can you compare the biological structure that has been distorted? The motherkoi seed, the motherkoi maize, and the maize that is not made into motherkoi. Do they have the same way that they can last? Can you compare how long can you store motherkoi and how long can you store the maize? Will they last the same time? Will they get spoiled at the same time? Basically, that is biological structure. And microbial constraint, basically, you're looking at some of the common antimicrobial inhibitors. For example, if you look at the lemon, it has acid. Citric acid is antimicrobial in nature. So that means that lemon may be able to prevent some microorganism from attacking it compared to other fruits that don't contain so much acid. So when you're looking at the nutrient content, you look at how much protein does this food contain, how much carbohydrate does this food contain, how much water does this food contain, and if it has all these constraints, does it mean that it may sustain, will it be able to sustain some of the microorganisms? One big example I'll give you is the difference between uh, the of course, the difference between an egg and, let's say, maize. They have the same type. Are they having the same capability of being spoiled? So basically, that is what I can say. I want you to read on oxidation, moisture, and of course the pH. So I will leave most of the things for you. Extrinsic parameters. Those are those properties of storage environment that affect both the food and the microorganism. For example, temperature, relative humidity, and then atmosphere. Temperature retards microbial growth, but when temperature is moderate, then it might actually affect the way that food is going to last. I will not want to dwell so much on that because I think the explanation is on it. And maybe if you would want more clarification, I'll be able to talk about that later on. Incidence and type of microorganism in food. Incidence, basically, you're asking yourself, when do microorganisms go into that food? For example, fresh meat and poultry. You really ask yourself primary sources and route of microorganisms. That is when the microorganism can go into that meat. For example, the stick knife or the knife that you use to chop the what? The meat, the animal hide, the JT, the hand of the handler, containers, handling and storage environment, the lymph node. Remember, the lymph nodes have the bacteria. All the bacteria that have infected that animal concentrate in the lymph node. So what happens if you chop the lymph node and then you get that lymph node get in touch with the meat? That means that the microorganism can move from one part of the lymph node and go and spread to the other part of the meat. So it's very, very important that you be able to prevent this microorganism from spreading into this meat by making sure that all those primary sources have been taken care of. So basically, when you're talking about incidence and type of microorganism in food, basically you're asking yourself, how do microorganisms go into that food? How do they destroy this? How are they able to get in contact with that food? Where are they coming from? So for example, if I ask you incidences and type of microorganisms in, let's say, um, prepared food such as salads, incidences and type of microorganisms in salads. So then you ask yourself, how do those microorganisms go into that salad? Starting from the preparation, of course, starting from the harvesting, buying from the mama burger, and coming to the preparation by the you, and of course, whichever water you're going to use and utensils and storage. Those are ways that you might be asking yourself when somebody asks you, describe the incidents and type of organism. So incidences, basically, you are asking yourself, how do those microorganisms come into those food? What would be the ways that these microorganisms will come into that food? So let's go to the next topic. Let's look at microbial growth and food spoilage. Of course, food spoilage results from growth of microbes in food, and they alter food visibility and in other ways rendering it unsuitable for consumption. And it involves predictable succession of microbes, and different foods undergo different types of spoilage processes. Toxins are sometimes produced. For example, algotoxins may contaminate shellfish and finfish, and toxins such as mycotoxins from the fungi might destroy some of the cereals. You also have some toxins that can be released into some other type of foods, such as bacterial toxins, for example, toxin produced by bacteria. And I want you to look at the toxins that can be produced by bacteria as well, so that you understand basically that toxins can also be produced when food gets spoiled. So that means that consuming spoiled food can really be a big problem. So two categories, we can look at food illnesses. Food illnesses happens due to consumption of spoiled food or food containing harmful microbes or their products. There are two categories of food poisoning. For example, uh, food infection, food intoxication. So that might come up, might cause food poisoning. So food infection, of course, occur as a result of consumption of living microorganisms, and food intoxication occur as a result of consumption of microbial toxins rather than the microbes. So that is the two difference. So symptoms, of course, include vomiting, diarrhea, fever, fatigue, and muscle cramp. So how do you prevent foodborne diseases or foodborne infection? So food infection, microbes are transferred to the consumer. Food poisoning results from toxins consumption. Then we'll ask yourself, how do you prevent some of these problems? For example, food infection or food poisoning. That is a question that I leave to you as a student to go and research and read, and of course explain to me later on. So major foodborne infection diseases are so many, of course. I will talk about salmonellosis, listeriosis, shigellosis, plesiomonas that causes diarrhea, and of course vibriosis, of course, that's caused by Vibrio parahemolyticus, and maybe there are so many others such as cholera, uh, typhoid, and so many others. So of course it is a table that you can pause and read on it and look at it, and of course look at the incubation period, the organism, and major food involved. So we've talked about before about food poisoning, and then when we talked about food poisoning, we talked about we can talk about food intoxication, illnesses caused by ingesting of food with preformed toxin. And example of food intoxication are Bacillus cereus, Staphylococcus aureus, Clostridium botulinum, that causes botulinism. Staphylococcus aureus, I also want you to look at the toxins that are released by Staphylococcus aureus. I want you to look at the Clostridium botulinum toxin the toxin produced by Clostridium botulinum. I want you to look at the toxin that is produced by Bacillus cereus. I'll also want you to look at the mycotoxins. Maybe it might be in the later part of this presentation. Food infection is illnesses caused by ingestion of food with microorganisms. And the incubation period and severity of the disease in the food infection is determined by inoculum microorganism ingested. This one means that it depends on the number of microorganisms you've ingested. If you've ingested 1 million microorganisms and somebody has ingested only 10,000 microorganisms, and somebody else has ingested 1,000 microorganisms, the severity will be different. For example, somebody who has has ingested 1 million microorganisms will not have the same severity as somebody who's just consumed 10,000 microorganisms. Of course, severity will be higher with somebody who has consumed 1 million microorganisms. So, laboratory diagnosis of food poisoning. Of course, how do you diagnose food poisoning? Of course, one of the specimen is leftover food, vomit, and specimen. That's what we take. Of course, we can culture the specimen for microbial isolation. We can also do serological techniques for toxin cell isolation. And of course, treatment, they will depend on the causative agent. So, of course, you're lucky that I'm presenting the mycotoxins, and the mycotoxins are toxins produced by the fungi. We have the egotoxin, aflatoxin, fumonicin, and acratoxin. So I want you to pause there and you read around that and you look at the vehicle food for egotoxin, aflatoxin. What would be the vehicle food? What would be the vehicle food for fumonicin? What would be the vehicle food for acratoxin? Some of the toxic syndrome associated with marine algotoxin. Algotoxin are associated with most of the common problems, especially when it comes to consumption of seafood. Of course, you should be able to you will come across some of the common problems such as parasitic shellfish poisoning, neurotoxic shellfish poisoning, ciguatura fish poisoning, amnesic shellfish poisoning, diuretic shellfish poisoning, estuary syndrome. Some of these are common symptoms that are associated with algotoxin. Algae is an microorganism, it's an organism that normally survives in water bodies, and this is normally a common problem, especially when it comes to the consumption of seafood. So, how do you control food spoilage? Of course, removal of microorganisms, low temperature, high temperature, reduced water availability, chemical-based preservation, radiation as a way of preserving, killing the microorganism, microbial product based inhibition. And these are ways that you can actually prevent you can actually preserve food so that the microorganism can be kept at bay. I want also to talk about canned food spoilage organisms that may be a very big problem. Canned food is food that most of us will want to eat, such as meat that's been canned. And what causes mostly canned food spoilage is, of course, the bacteria and the fungi and so many other things. So most of the things that causes canned food spoilage is normally when talking about canned food, you're talking about an environment that is quite tight and that does not have oxygen, And most of these microorganisms that survive at certain situation are organisms that have can adapt in an anaerobic environment where there's no oxygen at all. So with the minimum amount of oxygen, of course. But let's look at the characteristics. Most of these organisms are a mesophilic organism. I want you also to read around what are mesophilic organisms. So they are putrefactive anaerobes. They are able to produce some gases. So that means that the canned food, if you see the canned food it has expanded. Imefura hivi. So that means that the organism is producing some gases inside. And of course, the bacteria or the fungi may be able to be butyric anaerobe. And then there's another fact that is acidic flat source. That means that the food that you're going to use, if you eat that food that's canned, is acidic in nature. So that is a warning sign that you should not take this food. And most of the food, the microorganisms that can cause some of this spoilage are lactobacilli, yeast, mold, and some of the thermophilic organisms. And of course, thermophilic anaerobe producing sulfide. Sulfide, it means that some of this food may have some very, very bad smell. So means that you have to use your sense. If you taste the food is very sour and the food is very smelly, then that means that that food should not be consumed. Major group of chemicals used in food preservation. I think this one you can pause this lecture and then you can have it there. When you're talking about radiation, radiation is a way of preserving food and can be used to preserve some of the packages, can be able to kill all the microorganisms on the packages and surfaces and containers that are used to preserve food. We have ultraviolet light, gamma radiation, beta rays that may be defined as stream of electrons emitted from radioactive substances. Gamma radiation is used for ionizing radiation. And this means that it helps to extend the shelf life or sterilize the meat, seafood, fruits, and vegetable. Ultraviolet radiation is used for surfaces of food or food handling equipment and does not penetrate food. So each one of these radiation method is used differently. We have microwave. Microwave is also used as a way of preserving food, and X-ray can also be used to preserve food or containers used to keep or machines that are used to package the food. When you're talking about food, some of the preventive method of food infection, food poisoning, mostly we just need to entail some of the suggestions that I have put here. Refrigerating quickly, washing hands, cleaning cutting boards left over to be avoided at all cost. Avoid home canned food, because when you're canning your food at home, are you sure of the sanitary standards that you're going to have? Major categories, I'll be a little bit faster, but I want us to talk about major categories and examples of fermented milk products. I think there are so many products that need to be fermented, and most of these can be facilitated by yeast or mold. For example, those that can be facilitated by yeast, of course, and those that can be facilitated by the bacteria are different. When you look at this table, I think you'll be able to understand how some of those methods have been able to produce different products out there in the market. Major type of cheese and microorganisms used in their production. This one as well, you can pause this one and read and understand how microorganism can be used as a major ways of producing different type of food, converting this food into this type of a food. For example, milk to yogurt and so many other ways. Of course, you can pause that on that one, and then you see meat and fish fermented products, of course, sausages, hams, and bologna, salami, sushi, katsubushi. All these are fermented products that are out there in the market, and all of us might have come across some of them. Other fermented food, of course, this is just trying to highlight the importance of microorganisms and different products that comes with fermentation. Of course, silages, also some of the animal feeds that can be fermented for feeding our animals. And microorganisms as food, and food amendments. Of course, this tells us that microorganism can be used for other ways. Of course, a variety of bacteria are used and other fungi are used as animal and human food sources. And probiotics, when talking about probiotics, so for example, some of the yogurts that you take that are very expensive, of course, have been enhanced by being loaded with some very important microorganism. And of course, they have what we call microbial dietary adjuvants, and microbes are added to diet in order to improve the health benefit beyond the basic nutritive value. For example, somebody who have used antibiotics for a very long time, they might have killed all the beneficial microorganisms. So they need to return those microorganisms back to the gut. And some of these microorganisms can be able to be given through food. That marks the end of our discussion for today. And thank you for your time and for having this listening to this video. I'll share this to all the platform on MLM for North Coast Medical Training College for my students on hostel catering and nutrition. Thank you and have a nice day.